So my name is Tony Purcell. I'm a Professor of Research at Monash University in sunny Clayton, Australia. My field of research is immunopeptidomics, which is the study of peptides involved in immune recognition. So I'm in the biochemistry department here at Monash University, and we're part of the Biomedicine Discovery Research Institute. And we have a major focus in infection and immunity, and also cancer. And immunopeptidomics is one of the pivotal techniques that underpins a lot of discovery research in that area. Because we have quite a large team, 30 people or so, a number of dedicated instruments for doing immunopeptidomics, we focus across the whole gamut of immune recognition. So allergy, cancer, and immuno-oncology, infectious disease, autoimmunity, everything. So immunopeptidomics is a field that was coined probably about 15 years ago. It's an area of research that has been quite niche until probably the last 3 or 4 years, and this has really been driven by interest in immuno-oncology and this whole mantra of neoantigen discovery. So these are the peptides that come from mutations within cancers. The field itself has really revolved around the use of high-resolution chromatography, separation of very complex mixtures of peptides. So we're typically working with mixtures of around 100,000 unique peptide species, and using the use of mass spectrometry very sensitively to detect and characterize the various peptides. The challenges are that you're working with peptides that are very closely related. So typically they have very similar length, they have shared sequence features, so they're probably, at least in my experience, one of the most challenging samples that we can work with. We also work with very small amounts of material, so more and more now with clinical biopsy material. So having the most sensitive workflows and the most sensitive instrumentation is sort of critical to be, I guess, successful and to get the depth of analysis that's required to identify, clinically actionable peptides. So the current projects we're working on in my laboratory focus around 3 cancers: triple-negative breast cancer, colorectal cancer, and melanoma. That's in the cancer area. We also have a large program of research that focuses on autoimmune disease, and in particular psoriasis, which is an autoimmune disease associated with skin, sort of bullous skin disease, and also type 1 diabetes. So I guess precision medicine is now a term that's used a lot, and it probably means different things to different people, but at least for me it's really the personalisation of medicine, and where there's an immune component to a disease, or where the immune system can be harnessed to actually help fight disease. For me, that's where immunopeptidomics is sort of sitting as a pivotal technique. So my dream, if you like it, is to really be able to take the technology forward and to have serious impact in some of these diseases. So for example, personalised vaccines in cancer would be, I guess, one of the end games for us. We have 2 Bruker timsTOF Pro 2s in the lab, and we use Aurora CSI columns on both systems. One is using a nanoloop 2 system, where we use a trap-a-loop protocol almost exclusively for immunopeptidomics analysis. The other system, we have an EvaSep 1, also with the Aurora CSI column, and we've been getting fantastic data with that system as well. Yeah, so we enjoy the PASEF workflow on the timsTOFs. I think in terms of absolute depth and sensitivity, that's been probably the most sensitive instrument we've had access to so far. Using the timsTOF and the Aurora, we've had a number of successful projects, probably almost in press now. So for example, a very large study looking at SARS-CoV-2 peptide antigens across quite a broad array of different HLA types in humans. We've also had a number of quite successful campaigns looking at tumour antigens and tumour neoepitopes where we've been working with very small amounts of material, things like pleural effusions from mesothelioma, colorectal cancer biopsies, salivary gland biopsies in autoimmune disease and so forth. So we've had a ZenoTOF in the lab since September 2021. We had early access to the instrument through a collaboration with SCIEX. And look, we've been very impressed with the instrument. It's been very robust and been particularly impressed by the ZenoTOF capabilities. So this is data independent acquisition using the xenotrapping function. And we've applied that broadly across typical proteomics types of experiments through to immunopeptidomics analysis as well. In terms of the synergy between the Aurora columns and the ZenoTOF, I think the way it's influencing our work at the moment really is in the scale of what we can do and the confidence moving forwards in terms of having robust performance. And that's robust performance within the lifetime of a column, but also we've found as we move on to the next column, the performance is very similar, and then there's very little alteration in our workflows that are required. Yeah, look, our experience with the development of the SX columns I think has been a very positive experience for us. Really think it's a very elegant design and very much plug and play technology, which is exactly what we need for that particular instrument and the applications that we have. Since adopting the Aurora SX columns, I think we've really noticed an improvement in the sharpness of the peaks and the number of IDs we've been getting from the samples. We've run a lot of different types of samples on the Aurora SX through various immunopeptidomics projects, but also quite a lot of XenoSwath analysis of signalling in different types of antigen presenting cells. So we have a big project looking at the different interferons and how their signalling differs during infection versus, I guess, a constitutive state. I think the biggest help we get from the SX column in these projects really is around the sort of sensitivity we get. So I think we're certainly seeing probably a 10 to 15% improvement in IDs typically compared to other columns we've used. And so that's why we're sticking with them. So the combination of the Aurora SX columns and the ZenoTOF really has improved nanoflow chromatography on that system. And the fact I think it was custom designed to fit into the OptiFlow source really gives us a lot of confidence in operation. And there's really minimal tweaking of the system once the column's installed. And I think that's a massive benefit when you're trying to get good throughput, reproducibility and robustness in the instrument. I think if you're a ZenoTOF user and you're interested in nanoflow chromatography, the Aurora SX columns are definitely worth looking at. We've definitely been very happy with them, and certainly I don't think we are going back to our previous solutions. One of the most exciting projects to me is a project where we're actually interfacing with a clinical trial with a company called Grey Wolf Therapeutics, where we have an assay that is really, I guess, looking at mechanism of action of a small compound in the immuno-oncology space, and we've had to really tweak the system to get the ultimate in sensitivity and specificity for a clinical assay. I think the challenges now are that the types of samples we analyse are much more complex. So we've gone from cutting out gel spots to having fairly good liquid chromatography and separation through now demanding even more. And with the sensitivity of the instruments, we can see a lot more as well. So I think the challenges are centred around, well, how much can you see and how much can you differentiate? So for example, with a clinical sample, you'll have, let's say, the human proteome. You may have on top of that genomic information about that particular sample you want to incorporate. And you may also have proteins derived from the microbiome. So suddenly you've got a much more complex and much more difficult analysis than you originally contemplated. And having the resolving power and the separation, I think, is critical to actually get the most out of the data. So I guess as proteomics has grown and evolved over the years, the big changes for me are the number of attendees, the number of students really, I guess, being very passionate in the area, and the fact that the technology is now so much more accessible to more people. We've gone from it being a very specialised and very technical field to now being something that many more people can actually access. And at least in terms of immunopeptidomics, my pet area, it's gone from a room with about 5 people in it to an auditorium that's almost packed full. So that's been kind of satisfying. Been some big initiatives in proteomics globally, and I think it's a pretty exciting time for us all. I've been a member of HUPO for quite a number of years now, and counsellor for the last 6. And I think the fact that that's a meeting that brings everyone together to share ideas is exciting. And from HUPO, there's been a lot of initiatives that have been released, and I guess I'm proudest of the Human Immunopeptidome project, which again is close to my heart. I think some of the new big initiatives, such as the Pi-Hub initiative coming out of China, are also tantalising and interesting. I think it's going to be very exciting to work with those guys to see what we can do at a global scale.